Hello, my name is Kyle Miller, and today I want to talk to you about the question of how did axonal elongation evolve? And just to give this a little bit of context, for the most part, I'm a classically trained cell biologist, and this work has been done in collaboration with Aaron Craig, who is a physicist. And kind of just to give a whole summary of the talk in brief, essentially what we're doing is we're using biophysics to understand various modes of cell migration, and then what we're going to do is try to link these different modes of migration together in a pathway that ultimately links cytokinesis with axonal elongation. Okay, so why do we care about how axonal elongation occurs? And the simple answer is currently the treatments for spinal cord injury and stroke are relatively ineffective. And the idea is that if we understand how neurons grow, how they evolved, ultimately this will help the development of better treatments for injuries. But before we talk about cells and evolution, I just want to talk about how objects move. All right, so actually just to start off here. So this is my favorite sort of picture of a neuron, and it's a scanning electron microscopy image. So in the back here, we have a neuronal cell body. This is where the nucleus of the neuron is contained. We have this long, thin process, which is the axon, and we have a growth cone at the tip of the axon. And during the process of axonal elongation, what happens is that contractile forces in the growth cone drive what's called actin retrograde flow. And what this does is it pulls the substrate backwards, and then this pulls the axon and or the cell forward. And a really good way to think about this problem, I think, is an analogy with how cars move forward. And there was an undergrad in my lab, Candace Rivers here, and one summer she helped me put together this drawing. But the basic idea is that as you're driving a car down the road, you have a tire, and what's happening is the tire is pulling the road backwards, and then this generates a force that pulls the car forward. So if we consider this sort of array of filaments to be the actin in, say, a growth cone, the idea is that there's molecular motors back here, mostly non-muscle myosin 2, and they're generating contractile forces. So we've got like a convergence zone right in here. What happens is this pulls actin filaments backwards, and as they interact with the substrate, it pulls the cell forward. And you can just imagine the growth cone or the cell is going to advance down the road. All right. So I kind of like to show this picture. It's kind of fun. This is a drawing that Darwin made. This is the very first evolutionary tree, and I like this. So at the very top, it says, I think, and you can see that he had sort of outlined this idea that there's this tree that links us all together. And almost 200 years later, we're playing really the same game, except this time, what I'm trying to do is I'm trying to link together the processes of cytokinesis, that is how cells divide, cell migration, and there's a couple of different modes, and then ultimately neuronal migration and axonal elongation. And generally kind of the approach that we're using is we're considering how the flow of materials occurs in these different cell types. And we're using biophysical modeling of this flow to try to understand the pathway linking cytokinesis, cell division, over here with axonal elongation. All right, so just to jump into things, so here is a side-by-side comparison of cytokinesis and a type of cell migration called ameboid migration. And down here at the bottom, we have a time-lapse movie of a C. elegans embryo, and this is just an unconstrained free embryo. And it's been labeled up with GFP labeled non-muscle myosin 2. And what you can see in the movie is that in the center of this single cell, there's a convergence zone. So there's a region where actinomyosin contraction is strong, and things are flowing towards the center, towards this convergence zone. All right. Up here at the top, we have an ameboid cell. And well, we have a Walker carcinoma cell, and it's undergoing a type of cellular migration called ameboid migration. And the way this works is this little cell has been put into a small channel, and you can see the walls of the channel at the top. And this compresses the cell in the channel. And then what happens is that in the cell, there's a convergence zone, much like with cytokinesis. And the idea is that actinomyosin is being pulled towards this convergence zone. And when that happens, because the sides of the cell are interacting with the sides of this channel, as the actinomyosin on the surface of the cell gets pulled backwards, this pushes and pulls the cell forward to drive migration. And I will say, so if you look at the top here, you can see it says the reference frame is the cell. So this movie has been sort of modified so the cell just stays in place during the entire movie here. And we can look over here and we have these two chymographs that show the flow of material over time. And I'm going to come back to these in just a second. But for those of you that aren't familiar, chymographs are a way of looking at time-lapse data that's really powerful. And the key idea is to think of a movie as a 3D object, so it's like a cube, and it has dimensions of X, Y, and time. And we normally view movies by looking at different XY slices in a sequence. Now, what happens with a chymograph is this three-dimensional cube is rotated on an axis. So now we have time on one axis and distance on the other. And what we're looking at is more or less a graph of motion. So objects that are stationary are going to have straight lines, and objects that are in motion are going to appear as diagonal lines. And if we go back and we look at the last slide here, we have two chymographs that show the flow of material inside this ameboid cell and inside the cell undergoing cytokinesis. And during the process of cytokinesis, essentially things are kind of stationary at the beginning, and then when cytokinesis begins, what you can see is that there are these little diagonal lines that indicate the flow of material towards the center. And I'll start up the movie here so just so you can see it. So these spots that are moving towards the center are represented by these diagonal lines. And I invert the colors because it's easier to see dark things on a white background. But and then in terms of the chymograph for ameboid migration, what you can see is in the uropod, which is the tail of this migrating cell, things are more or less sort of stationary in the cell frame of reference. But at the leading edge, you can see this retrograde motion. So things are flowing backwards. And really, when I sort of was like thinking of this problem, looking at these movies, it really struck me that if you look at the front end of a cell undergoing ameboid migration, it looks pretty much like one half of a cell undergoing cytokinesis. And since the molecules involved in ameboid migration, for example, myosin and actin, and the signaling pathways, for example, Rho in particular, are common between these two sort of cell types, it kind of leads to this idea that perhaps ameboid migration somehow evolved from cytokinesis. All right. So in terms of trying to understand this problem, the approach we have used is to do biophysical modeling. And I can take this same sort of data that we had before. So here is our C. elegans embryo undergoing cytokinesis. Here is our ameboid cell undergoing migration. And this time the chymograph has been just expressed in its sort of normal format. But in any case, like it's in the lab frame of reference. But in any case, we can trace the velocity profile of motion across these chymographs, and then put them together in just regular graphs. And this shows a graph here below. So this is cytokinesis, and we have the velocity over here on the y-axis and distance from the convergence zone on the x-axis. And what you can see is that in terms of velocities, stuff on the left moves towards the center, stuff on the right moves towards the center. And the cells undergoing cytokinesis are stationary in time. Now with ameboid migration, what we have here is that the stuff on the left-hand side, that is the tail of this migrating cell, is advancing forward. And the stuff on the leading edge of the cell is undergoing a little bit of net retrograde motion. So the tail end is moving forward at about 8 microns per minute, and the front end moves back at about -1 microns per minute. But the really striking thing is that when you look at these two processes side by side, you can see that there overall is a very similar sort of velocity profile. And in terms of understanding this, the way that they really differ is that in the ameboid cells, we have the nucleus of the cell right here. And kind of the idea is that it is sort of this large, relatively incompressible structure, and it kind of creates a bulk that helps the cell press up against the sides of the channel. So when we have this flow backwards, then the cell is pulling the channel backwards and it's moving itself forward. And I will note the traction forces here are very small. You can kind of think of this like the tanks of a tread, the treads on a tank. So when they're pulling against the ground, the traction forces actually are really very small. It's almost as if, the cell is rolling across the substrate, generating a very sort of minimal amount of traction force. Okay. In terms of modeling the cellular flow, we use what is called an active fluid approach. And there's a couple of assumptions there. Essentially, at the time scales that we're interested in, cells more or less act as fluids that are able to generate forces internally. And this is the governing equation that describes cellular flow. And it's really just it's a force balance equation. And in brief, what it's saying is that we have some profile of internal force generation. So some sort of description of the change in the motors over change in distance. We also have some profile of adhesions with the substrate. So what we can imagine is that there's going to be frictional interactions between the cell and the substrate. And the forces that the friction generates is going to be proportional to the velocity. So if the velocity is faster, forces is going to be higher. If the adhesion is stronger, then forces are going to be higher. And this third term in the center talks about the deformation of the cell. So when you have convergence or divergence, so when things are flowing together in a cell, it's deforming the cell, it's changing its shape. And this term relates viscosity with the velocity of motion to tell you about the forces that are involved in deforming the cell during the process of migration. And the approach here is relatively simple. So what we do is we have our experimental data shown, for example, on this plot as blue points. And then what we do is we go in and fit this model to the data. And from that, we can back out our traction forces, we can back out the distribution of motors in the cell and the viscosity of the cell to put together a mathematical model that describes both of these. And in doing so, the equation suggests the main difference between cytokinesis and ameboid migration is the adhesion profile. And very simply, that is that in cytokinesis, adhesion is symmetrical on both sides, and ameboid migration, adhesion is asymmetrical. So the cells just interact on one side with the substrate. And you could see that in the movies. Now, in terms of how ameboid migration evolved, a hypothesis that we're proposing is that perhaps mutations activated cytokinesis during interphase. And so if you kind of think about it, so here's a little cartoon of a dividing cell, and you can imagine there would be normally like two nuclei in the cell. So now we're past telophase, we're into the process of cytokinesis, and you can imagine everything's symmetrical. But imagine, if you would, if you had a cell that was at some stage in interphase, and it just has a single nucleus, but this whole process of cytokinesis is engaged. Now, what's going to happen is you're going to have a contractile ring on one side in just a single nucleus. And because this single nucleus makes one side larger than the other, then this side is going to be able to press up against the sides of a channel. And you can see these little green sort of like squiggly lines. These are adhesions over here. So as actin flow moves towards this convergence zone, it pulls the cell and pushes the cell forward. Okay. Now, in terms of, there's more than one type of cell migration, and very often, especially like in the context of cancer, there's a lot of discussion of ameboid cell migration, and that's what we started with, really to make this analogy with cytokinesis. But through our bodies, mesenchymal style migration is very common as well. And really the main sort of difference is that ameboid migration occurs in 3 dimensions and mesenchymal migration occurs in 2 dimensions. And this is a fish keratocyte. It was injected with rhodamine phalloidin, so we can track the movement of the actin filaments. And when you look at the pictures of the movies of the cell, they look dramatically different. So over here, this is sort of like a long cylinder, and these mesenchymal cells look very much like a fried egg. The thing that's really striking is that when we look at the chymographs, they look very similar. So in both cases, we've got the rear of the cell, and this advances forward as a whole. And that you can see is these sort of parallel diagonal lines. And at the leading edge of the cell, what's happening is actin filament assemblies occurring at the very leading edge. It forms interactions with the substrate. And then because there's a convergence zone sort of right in front of the nucleus here, and actually kind of around and behind the nucleus here, the stuff at the leading edge gets pulled backwards. And in both cases, one, I think, really straightforward way to think about this is that these forms of cellular motion are very similar to the way that tires interact with the road. And once again, the bottom of the tire is pulling the road backwards, and then it pulls cars forward. So actin retrograde flow pulls the substrate backwards, and then this pulls the cell forward. In terms of the modeling, if we look at the velocity profiles, you can see that once again, they're strikingly similar. There's a little bit of a shift of the position of the convergence zone between these 2 types of migration, but overall, the modeling suggests that the mechanics are very similar. And in terms of how mesenchymal migration evolved, so one sort of question is, well, which came first, ameboid migration or mesenchymal migration? And at this point in time, it's really hard to say. I will say that it is well accepted that many types of cells can switch from ameboid to mesenchymal migration when there's either alterations in Rho activity or changes in adhesion. And an idea that's been proposed is essentially that these 2 forms of migration are very similar, and they reflect more how cells interact with 2 and 3 dimensional environments as opposed to a dramatic change in terms of the identity of a cell type. Okay, so with this sort of background, we've talked about cytokinesis, ameboid migration, mesenchymal migration. We now kind of want to get into the question of how when neurons first evolved. And just to kind of lay this out in sort of broad strokes, neurons are only present in metazoans. They're only present in animals, but not all types of animals. So in this little tree, we have a couple of different names for people that aren't evolutionary biologists. So these peripher would be sponges. We have what are called jellycombs, which look an awful lot like jellyfish, but in fact diverged long ago from that sort of lineage. There's a very simple organism that has 6 cells and no neurons. And one idea is that it initially evolved from some sort of species that had neurons and lost that over time. And then, of course, we have bilateral animals like ourselves that obviously, well, do have neurons. And the question is, like, how did neurons evolve in metazoans? And in order to sort of like get at this, what I'd like to do is kind of make this bridge between cellular migration and neuronal migration. And I think the most straightforward way to do this is just to show 2 movies. So over here, we have our movie of a mesenchymal cell undergoing migration. And over here is a movie of a migrating neuron. And it's a little bit hard to see, but we've got a neuronal cell body at the end, and then there's a long, thin axon, and then a growth cone at the tip. And hopefully what you can see from both the chymograph and the movie is that at the very leading edge of the cell, the growth cone is exploring the environment, it's making adhesions with the environment, and then there's convergence zone at the tip. And during the process of migration, the cell body at the back moves forward at a relatively constant rate. The materials along the length of the little neurite are moving forward as well, and the growth cone is advancing. And the thing that I really want to pull out or point out to you is that in both cases, the bulk of the cell body, the nucleus, these are all advancing as a coherent unit. And it's retrograde flow at the very tip of the cell, or the leading edge of the cell, that's generating these forces that drive migration. Now, the question is axonal elongation a form of cell migration? And one thing is, I just kind of want to point out that for a long time, people thought that the way neurons elongated is that microtubules were stationary along the entire length of the axon, and that microtubule assembly in the growth cone drove axonal elongation. And this is mechanically, this is a very sort of different process. When we look at these 2 things side by side, I think it paints a very different picture. So once again, here is a movie of a neuron undergoing neuronal migration. And this is a time-lapse movie tracking the motion of docked mitochondria that shows the motion along the length of the axon during elongation. So in both neuronal migration and axonal elongation, we have a growth cone at the very tip. And then once again, we have a long, thin axon. And if you can just watch the movie for a moment, and you can see towards the front, we have a growth cone in both cases, and they're both advancing. And then when we look along the length of the axon, we can see that material along the length of the axon is also advancing. And the one place, or the one sort of major difference, is that during axonal elongation, the axons grow, they lengthen. And by looking at the velocity profile, and you can see this in this chymograph, though it's really focused on the braid tip, what happens is that the growth cone is advancing faster than stuff along the length of the axon, which indicates that the axon is stretching. And if we look at the flow patterns, so these are data from a couple of different experiments that have all sort of been combined, and then we've done mathematical modeling of neuronal migration and axonal elongation. And in neuronal migration, what happens is the velocity of material is more or less constant along the length of the neurite, and there's retrograde flow that's happening in the growth cone. And during axonal elongation, the stuff over the distal end of the axon, so if you notice this scale goes from like 0 to -40, and this goes from 0 to about -200. At the tip of the axon, you see forward advance in a pattern that's very similar to neuronal migration. Nonetheless, towards the cell body, everything along the length of the axon, well, the bulk of materials along the length of the axon are stationary relative to the substrate. And the idea is the cell body is staying in place and the axon is lengthening by stretching. And this suggests, in terms of our modeling, that really the big transition from neuronal migration to axonal elongation is a difference in adhesion under the cell body. So during the process of axonal elongation, in this little cartoon, I've drawn a bunch of adhesions. So in axonal elongation, the idea is that these adhesions are going to be relatively strong. And then during neuronal migration, the idea is that there'll be fewer or weaker adhesions and fewer and weaker adhesions along the neurite. And what this does is it allows the forces generated in the growth cones to pull the substrate back and to pull the cell forward. And before I move on and start talking a little bit more about evolution, I just want to say that yes, the processes of axonal elongation, neuronal migration, cellular migration are far more complex than what I'm going in here. Really, our intent is just to develop a general model that we can use for all of these different forms of motility. And in that way, we can start thinking about sort of big picture ideas about how cytokinesis might be related with axonal elongation. So just to talk a tiny little bit about how the evolution of neurons occurred. A lot of this is really common sense sort of thing. So we assume that sometime early in the evolution of eukaryotic cells, these types of cells were capable of undergoing actomyosin-based cytokinesis as we do. And then at some point in evolution, when eukaryotic cells were single cells, they hadn't formed multicellular organisms, the cells acquired the capabilities to migrate and explore the environment around them. And only much later, so if we look at this tree and it's roughly scaled, well, it's scaled in terms of genetic distance, but it's also roughly scaled in times. You can see that the entire branch of animals, that complicated bit, a graph that I had shown you, is all in this tree. And it's somewhere in here that axonal elongation happened. Unfortunately, these events occurred, well, roughly 2.5 billion years ago, and then these events up here occurred maybe 500 million years ago. But the issue is the fossil record of when the first neurons evolved and the sort of early events in eukaryotic cells are poorly preserved. And just because of the immense amounts of time, it's really difficult to construct genetic sort of like, or understand the sort of relationships between different species. And there's a really nice article by Lammer et al. From 2019 that I think does a really great job in explaining one of these big controversies. So as of now, it's really unclear how the various sort of branches in metazoa evolved in the order. And if you go back to the older sort of understanding of this problem, for a long time it was thought that initially a simple sort of species like sponges evolved, and then and sponges have no neurons, but they do have neuron-like cells. And then later, more complicated types of animals arose, and at least in one case, one lineage lost the ability to grow neurons. But when analyzing the genomic data and putting together the phylogenies that construct this, in fact, it's very easy to, or it's very common to find that ctenophora, this is the comb jellies, which do have neurons, seem to be at the very base of the tree. And this sort of suggests that, in fact, sponges lost the ability to form proper neurons, as well as the placozoa. So this would suggest that losing the ability to have neuronal cell types occurred either once or twice during the process of evolution. In both cases, we're still sort of at this interesting question, and I'm, in a sense, it might seem like I'm dodging the question, of how the first neurons evolved. But at this point, we're not even quite sure of the proper sort of lineages that gave rise to the evolution of nervous systems. And the simple suggestion that we're making here is that by analyzing flow patterns, ultrastructure, and gene expression in ctenophore neurons, these would be the comb jellies, and comparing that with the expression of genes and the flow patterns in, for example, mammalian neurons, we can start to get a better understanding of the process of evolution and ultimately put together a really tight understanding. And in this little diagram, so these arrows indicate tentative evolutionary pathways. And so, for example, we can imagine from cytokinesis, ameboid migration may have evolved first, and then mesenchymal migration evolved later. And likewise, from mesenchymal migration, one might sort of suspect that neuronal migration first evolved, and then later axonal elongation evolved. Nonetheless, there are a lot of different ways that this could have occurred. So we feel like this is a really interesting question that hasn't been explored, and there's a great deal of really interesting biology that I think is going to be uncovered by understanding the evolution of axonal elongations. So I'll just go ahead and quickly acknowledge a few friends and collaborators. So Steve Heidemann was really helpful in providing feedback on this manuscript, and really fun to talk to about the problem of how axons evolved. Anthony Cognata is a friend of mine who studies evolutionary biology, and he gave an earlier version of the manuscript a good read and helped me out quite a bit. And Aaron Craig at the Central Washington University is the one that did all of the modeling in this talk. And of course, I appreciate the support from the NSF and the NIH and various other funding sort of agencies or organizations here at Michigan State University that have been so supportive. So thank you.